Well, good afternoon. It's great to see you all here. We have another great program, thanks to the Dana Foundation. And I won't begin by trying to even characterize, let alone discuss research about autism and people with autism. But I will say this is going to be a fascinating discussion, and I think you're here for something really special. I'm Rush Holt. I'm the CEO of the American Association for the Advancement of Science. We're, as you may know, the world's largest general science membership organization with members in, I think about 80 countries. So although American is in our name, we are very much an international organization. We do things to promote communication among scientists and between scientists and the public. And we have programs in science and public policy, science and society, science and religion, science and the law, science and human rights, science education. And we are quite active speaking out on behalf of science and speaking out to where science can be brought to bear on public issues. Just out of curiosity, let me ask, how many members of AAAS do we have here? Great. Well, those of you who aren't, it's easy. It's not expensive. You get your own copy of Science Magazine each week, either digitally or hard copy. I prefer hard copy, but they do pile up fast because they come every week. I would say further, not only will you find it interesting and rewarding to be a member of AAAS, it helps us. We have 120,000 members. And as we advocate for science and for the good application of science in public, on public issues, it helps to be able to say, we have all of these members. So you help us if you join also. It's pennies a day. So anyway, but this is courtesy of the Dana Foundation tonight. And with an introduction of our really outstanding panel, I would like to ask Deborah Runkle from the AAAS Scientific Responsibility, Human Rights, and the Law program to introduce the speakers. Thanks.
Thank you, Dr. Holt. I would like to add my thanks to the Dana Foundation. They have been sponsoring this series on neuroscience and society for many years now. I do have to tell you, I regret to tell you that this is, for a while at least, this is the last in the series. And I know I can see a lot of familiar faces. I know a lot of you have come to many of these before. I want to introduce to you the man who's going to be the moderator for tonight's program, Dr. Barry Gordon, who's the professor of therapeutic cognitive neuroscience and a professor of neurology with a joint appointment in cognitive science at the Johns Hopkins School of Medicine. So if I can call on Dr. Gordon.
Thank you, Deborah. Thank you all for coming. I'm going to be very brief in my comments. The purpose of this public forum, sponsored by the AAAS and the Dana Foundation, is to try to present some of the current scientific thinking about the basis or bases for autism and about possible therapeutic or known therapeutic efforts for autism. And I'm here because of an accident of birth, because of our son's birth. My wife, who's over there, and I have an autistic son. He's what's called low functioning, nonverbal, etc., and he's now a young adult. And so I, like I suspect some of you here, have a real direct personal interest in these kind of issues. And what we have today is we're going to be discussing with Dr. Daniel Geschwind the genetics, plus possible therapeutic implications of the genetics, with Dr. LaSalle, the epigenetics, and with Dr. I always call you Dr. Dawson, Jerry, some aspects of therapeutics. But what I'm going to do first is tell you a little bit about the definition. I think many of you are aware that autism was and is still currently defined by behaviors. There's no biochemical marker, no objective signs. It's still at the behavioral stage at which it's defined. And I think you're also aware that when we talk about autism, we're actually talking about something that is, I hate to say, but a little nebulous, because the criteria that have been used have varied over the years, ever since it was first given a name, a definite name in 1943. And the definitions now also vary. So this is, by the way, my personal opinion. That's why the disclaimer slide was up there. Even currently, there's the way autism is used in popular usage, and I won't elaborate on that. There's the way it's been used in education and medical practice, where, again, it's not necessarily in keeping with some of the formal definitions. In clinical psychology and psychiatry, the Diagnostic and Statistical Manual 5 is now the, quote, official rulebook for diagnosis of autism, and I'll show you some of that definition. And in research, not only is the DSM-5 perhaps used, but in addition, there's the Autism Diagnostic Observation Schedule, the second edition, by Kathy Lloyd Rutter and others, that are used as the gold standard for defining autism. So there's a number of different definitions floating out there. And one of the things I would caution you as public or scientists or whatever is whenever you read about autism, you might want to dig at into what definition people are using. The one used at the DSM, the current DSM, has several criteria. And for speed, I'll just go over it because you can read faster than I can speak. Persistent deficits in social communication and social interaction, all right? Restricted and repetitive patterns of behavior. The symptoms must have been present in the early developmental period, but may have been masked, may not have been appreciated for a long time. And the symptoms cause clinically significant impairment, and these disturbances are not better explained by intellectual disability or global developmental delay. And that's the literature cited on this. With that definition in mind, partly in mind, our speaker is going to be telling you about autism. And I will just introduce first Dr. Daniel Geschwind. And in the interest of speed, thank you. In the interest of speed, you can read his biography. Every speaker has 15 to 20 minutes. All right, I'll hold them to that, to about a millisecond, give or take. And all the questions keep to the very end, but there will be time for questions. Dan, my pleasure.
So I want to thank the Dana Foundation and AAAS for giving us the opportunity to tell you a little bit about autism and engage in a dialogue. And obviously, I wouldn't be talking about genetics of autism if there were not a substantial contribution from genetics, but I'm going to go over that now, and hopefully we can discuss it. So the outline is, we're in the midst of a genetics revolution in a way, and I'll describe that very, very briefly. The notion of personalized medicine, precision health, it applies to autism just as it applies to any other disorder. In fact, perhaps even more. As Dr. Gordon said, this is not a disorder that's explained by etiology. It's described, it's a syndrome that is based on the description of behavior and cognition. So, similar, excuse me, I'm okay, to something like cancer, it's we'll do better if we can define it more specifically. That's the notion, based on etiology. I'll tell you what genes have been identified to this point and what they're telling us, what they might mean for clinical practice and treatment, although we're at very early stages. So a lot of what I'm going to say is talking about the promise. And here are my disclosures. I provide scientific advice or collaboration with these various commercial entities there. The only one that's really directly related to this is Ovid Therapeutics, which is a small biotech company trying to make therapeutics for rare diseases. So the goal of precision medicine, I mean, you've heard this all before, deliver the right treatment every time to the right person. And if we have a syndrome that is defined by cognition and behavior, we don't know what the cause is, we really have a conundrum. And of course, I'm not saying prediction is easy. Prediction is very difficult, especially if it's about the future. But what we'd really love to be able to do is to not just predict, but be able to prognosticate and choose the right therapies based on specific genetic diagnoses. That's the goal. And I mean, if you think about it, I see a lot of people wearing glasses, and some who aren't wearing glasses probably had LASIK or are wearing contacts. You wouldn't all wear the same pair of glasses, right? That sounds ridiculous. But essentially, a lot of what we do in medicine is give everybody the same pair of glasses. So essentially, there are two major drivers of this: decreasing DNA sequencing costs and computer power increasing. So this gives us an enormous armamentarium of big data and genomics with which to explore disease. So one of the things I wanted to introduce you to is, what is the nature of genetic variation, rare versus common genetics? And I'm basically going to put a ton of packed information into this one slide. Basically, if you think about it, genes that, genetic variants that are common in the population are from a common ancestor, usually thousands and thousands of years ago. The things that are in 1 to 5% of this population in this room, that's common genetic variation. And there are not going to be too many highly deleterious genetic variants in that, because evolution has acted to remove them from the gene pool. And so what we see is that the high frequency variants have small effect sizes with regard to their effect in terms of producing risk for a disease or a disorder or a syndrome. And rare mutations like genetic syndromes like Fragile X or Down syndrome have large effect sizes because you just need that one gene, but they're not common in the gene pool. And in fact, many of the things are genetic but non-inherited. So it would be like Down syndrome, where there's a genetic mutation, but that's not inherited from the parents. Parents don't actually have it in their somatic cells. It's just something that happened in the germline. So what I'm going to tell you is essentially, and I'll just boil it down to a few sentences, is that autism risk is a combination in the population of both rare and common genetic variation, but that combination varies from person to person. And I'll show you that in a bit. So it's not one or the other, it's both from a population standpoint, but individual, it may be one or the other or an intermediate combination. So what we've learned from about now 15 years of really genetic research that was really, I would say, pushed forward because of families and because of two major organizations, but family, it was a partnership between these advocacy organizations and researchers. And the first genetic database was the AGREE, which Autism Genetic Resource Exchange, and then a lot of the other recent work is from the Simons Foundation, Safari. So essentially, you can have these de novo genetic variations, that is, acting dominantly but not inherited, and this is a bunch of genes. You could have autosomal recessive, which is a very small percentage, but definitely occurs. Those are some examples. You could actually have autosomal dominant inheritance that actually is inherited from the parent. Or you could have things that are X-linked, like Fragile X and Duchenne muscular dystrophy. And then we have what is the additive common genetic risk, where it's a panoply of very small effect risk factors from both mom and dad. All of these modes of inheritance are known to occur in autism. And actually, as it turns out, genetic testing can currently identify about 20% of mutations occurring, contributing to autism. In other words, if we have 100 children who come to clinic, we can, it depends on how the subjects get there, how they're ascertained, but somewhere between 15 and 20% will, so that we do clinical microarray, Fragile X testing, exome sequencing. We actually now can sequence the whole exome quite effectively and very inexpensively. So what's happened is, this was a review that I wrote with a former postdoc, Brett Abrams, in 2008 in Nature Reviews Genetics. There were just a handful of genes, mostly syndromic forms like Fragile X, little understanding of mechanism, and not just in autism, but in a lot of major neuropsychiatric disorders, pharmaceutical companies, because of their lack of success, were withdrawing from CNS research. This is a paper from 2013 But if we kind of go circa 2016, more than 200 candidate genes with different levels of evidence for them, much clearer mechanistic models beginning to come above the surface, many advances, evidence for convergence and cross-disorder overlap, which I'll describe to you in a second, what I mean by convergence, and beginning of major drug development efforts in autism ongoing, because just like identification of mutations in cancer, once you have a cause, a clear mutation, there's something that you can begin to try to target and reverse. And so now, what I'm showing on the left are all these rare syndromic forms of autism. Some of them are inherited, most of them are de novo. This is just a beginning list. They account for about 4% of autism. That currently, we expect that as we keep discovering more and more of them, there'll be about 20%. And that's shown there. Then we have all the way on the right, we have about half of it is from predicted common inherited variation, and about 40% is unaccounted for, giving room for environment or other rare genetic variation. Does that make sense? To some degree? So we have many, many genes are predicted now from this gene finding. We haven't found that many, but it's predicted there might be more than 1,000 that contribute. None account for more than 1% of cases in terms of these rare mutations. You can see the percentage shown there. Even if you took the 10 most frequent forms, they would be less than 1%. Fragile X is probably the most common, and it's 1 to 2%. But if we look at those that are discovered by genome sequencing, the top 10 add up to less than 1%. They're highly additive effects, so you might have a rare variant that predisposes you, and on top of that, common genetic risk. And there's strong pleiotropy. That means that these genes aren't specific for autism. They disrupt brain development, predisposing and enhancing risk for a number of neurodevelopmental disorders, including autism. That's why there's a lot of comorbidity with epilepsy and intellectual disability. So knowing the genetic basis of the disorder has significant implications for treatment and recurrent risk. If you find these new de novo mutations, right, it's like having Down syndrome, the recurrence risk for the family is much less than if it's an inherited mutation. Knowing how it's inherited can also help the counseling, if it's recessive versus dominant. But from my perspective, it provides hope for the future in terms of identifying the mechanism of disease, and over the next decade or so, targeting therapy towards that. It's a long road to hoe, but that's our hope. We might identify gene-environment interactions, the actual mechanism by which the mutations might occur might be preventable, and of course, early diagnosis and intervention might be allowed. So one of the concepts that we started with and that one has to appreciate is that autism is on a continuum with normal variation to some degree, and overlaps with other neurodevelopmental disorders, so that the risk, autism, the core features, and originally language was part of the core features. This is from 2007, and now it's just focused on the social behavior and repetitive restrictive behavior. But if you move into the red zone on all three of these features, you're at high risk for autism. But the common genetic variation isn't just pushing you towards autism, it's modifying language, social behavior, repetitive restrictive behavior in the general population. And so it could be that there are environmental factors interacting with mutations or small effect size individual polymorphisms that are interacting that kind of push on the teeter-totter, pushing you over on these normal distributions into having risk for autism. And this is just some data to show this very recently. This was more of a prediction in a model. This is data from Elise Robinson, where they're basically showing, if you look at we have genetic correlation and then a couple of different categories on the x-axis. And basically what it's showing, the first two are taking composite polygenic common risk for autism. You basically add it all up and weight it and ask, how does that relate? Can that predict social cognition in the general population? And this is done in a large English cohort. And there are two different scores, one from a Danish cohort of 14,000 children and another from something called the Psychiatric Genetics Consortium. But they both have almost a 0.3 correlation the genetic risk for autism with the modification of social cognition in the general non-autistic population. So the challenge now is that we're finding a lot of genes. They provide targets for mechanistic understanding and therapeutic development. Some have small effect sizes, some have large effect sizes, but they highlight extreme genetic heterogeneity. If I have 100 cases of autism, I might have 100 different forms. Will we have to develop a specific treatment for each disorder, or will there be convergence in specific pathways, developmental stages, or brain circuitry? Again, the analogy I use is cancer, radiation to fight proliferation. Not a great therapy. I'd rather target the individual mutation, but better than nothing. And so when I think of convergence, I think of levels. And as a neurologist, we start by thinking that behavior comes from specific brain circuits. And so therefore, the social aspect, which is really the core of autism, and the restrictive repetitive behavior, or alterations in mental flexibility, really have to involve brain circuits that are frontal striatal, frontal temporal, frontal parietal. That's just a kind of cartoon way of thinking about it. But at some level, there has to be convergence at that level. You have to affect the parts of the brain that are involved in that, in that function. But from a molecular standpoint, I've just listed 3 different molecular conditions that increase risk for autism. So the question is, are these going to converge on molecular pathways, biological pathways, brain regions and circuits we just discussed? So a lot of the work that's been going on in the field now, we're really challenged to ask, is there convergence? And so one question we asked is, where and when do autism genes act? Do they act after birth? Do they act before birth? Now that we have hundreds of them, we can actually ask this with quite a bit of power. And what I'm showing here is embryonic development up to birth, then postnatal development. And these are all the neurobiological processes that are ongoing on. And in fact, in the middle of gestation is when the cerebral cortex is developing and neurons are being born and migrating, and the whole circuits are setting up. And that, it turns out, is where most of autism genetic risk lies, prenatally. If I ask when and where are these genes expressed at their highest level, it's prenatally, and it's in, I'll show you, basically 2 major categories, and this is an oversimplification. One of them are things that are involved in the birth of neurons that regulate what's called transcription or cell fate, that are involved in the birth of the neurons, the cells that are signaling in the brain, and that's very early, probably before 16 weeks, and then synaptic development, the development of neuronal connectivity. And those kind of cross and are around 16 weeks gestation. So this is happening prenatally. And what's interesting is that there's a predilection, it's not 100%, but there's an enrichment in hitting neurons that are in the superficial layers of the cortex that connect regions of the cortex, both between hemispheres and long-range connections across the cortex, connecting higher-order association areas in layers 2, 3 predominantly, and a little bit in 4. And that's shown right there. So there's convergence in early development and then the development of cortical connectivity at the genetic level. That's where the genes are acting. Not all of them, but there's convergence there. It's very non-random. How about in the brain? Is there a shared pattern of pathology? So we used molecular measures to measure the RNA, and people have looked at protein in the brain. And we're finding the same thing. What I've shown there is a clustergram, where on the top we've clustered post-mortem brain from cortex of patients with autism red and controls black. And you can see the red are largely, not 100%, clustering together. And what that is showing is that they have a shared pattern of the dysregulation of gene expression. 2/3 of the cases show upregulation of genes in very specific pathways involved in these glial cells, which are supporting but critical for the development of synapses. It's called neuroinflammation, but it's inflammation. It's inflammation, but we can discuss that in more detail. There's parallel downregulation of neuronal genes involved in vesicle transport and synaptic signaling. And the causal genes, if we ask where does the genetic risk lie, it's primarily in the neuronal component, so that we think that there's neuronal dysfunction and then subsequent upregulation of this compensatory, potentially maladaptive glial response. These findings define a molecular pathology of autism. There have been several studies, and now over 100 cases and controls studied, and this finding is continually reproduced. We also, that's at the level of, so we talked about convergence at the level of the genes that are identified, at the level of the postmortem brain. Some of these genes converge on biological processes, and so you can think of, I'm just going to there's a notion that some of these genes are involved in the balance of excitation and inhibition. I mentioned to you the genetics shows abnormalities in the neurons that are connecting regions of the brain. The imaging studies show deficits in functional connectivity as well. But we can begin to target specific pathways, and these are just a bunch of them, where there's non-random piling up of genes that are identified affecting neuronal activity, neuronal cell adhesion, which is important for connecting the neurons, as well as changes that occur. We know that the brain is plastic and synapses change with learning over time. And this depends upon activity-dependent protein synthesis, and a number of the genes are involved in this as well. But how do we develop therapeutics now? We have some clues. There are multiple avenues that we could actually be going down. How do we do this? It's been standard to use animal models in therapeutic development. That hasn't been vastly successful in the neuro field. But one of the huge advances came with the ability to make stem cells from somatic cells, take a patient fibroblast, create an induced pluripotent cell from which you can make any cell in the body, make neural progenitors, and grow patient-specific neurons. And now this has gotten quite sophisticated, and we can really grow little forebrain spheroids or mini-brains in a dish. This is just one of the first papers where we collaborated with Ricardo Dolmetsch when he was at Stanford. This was driven by Sergio Pasca to actually use these. This was the first syndromic form of autism, this form called Timothy syndrome, which affects a calcium channel. So the notion is, if we look at human relevance, mechanistic insight, and the ability to test a lot of hypotheses, that is, throughput, these iPSCs and primary human neural progenitors are really at the top there. And I think they are going to, there's a lot of promise there, but it's just promise at this stage. So another point I want to bring up, and then I'm going to end, is that we can discuss this, but a number of neurodevelopmental disorders that have been modeled in mice can be reversed in the adult versions of these models. And should these findings be generalizable to humans, genetically identified pathway therapeutics could become an important area. That's just some guy named Geschwind saying that. Here is many of you have probably read his book, but this is from Paul Kalanithian, who was a neurosurgeon at Stanford, who was given a diagnosis of lung cancer at age 36, and he's written a remarkable memoir. But this was from his Sunday review in the New York Times. And then my health began to improve thanks to a pill that targets a specific genetic mutation. This was not curative, but it did extend his life. He eventually succumbed. But this is the notion that we're looking for, to try to target, with much more specificity, and I think that's the hope for the future. So in conclusion, autism risk has a large genetic component, but its etiology is multifactorial and heterogeneous. Genetic studies have identified many dozens of risk genes. We can capitalize on these findings using convergent evidence from studies in human brain and model systems to begin to elucidate the neural system's basis of autism. I've described a little bit what we know so far, but it's just the tip of the iceberg. And that these genetic findings provide a starting point to create in vitro and in vivo models to identify potential convergent molecular pathways to development of targeted therapies. I just put this up because my work and all the work in the field that we do depends on our funders, but also I just go around and give talks. These are the people who actually do the work in the laboratory. Thank you.
Thank you, Dr. Geschwind, and thank you for being on time as well. Dr. LaSalle, Dr. Legine LaSalle will be talking to you about, well, what she's going to talk about. And there's a phone up here. Okay.
Great, I'd like to start by definitely, so which, is it all of these probably work? Okay. Love to start by thanking the organizers for the invitation. Today I'm going to be telling you about our approaches in the field to solve this very complex puzzle of gene by environment interactions in autism. And I'm specifically going to be talking about how we're using the womb's strange epigenome to get at that. But I want to start with this title, Beyond the Four-Letter Code, thank you, to drive home the concept that the DNA sequence itself is not enough to explain some of the complexities in autism etiology. And to bring this concept home, I'd like to start with in the realm of science fiction before we get to the state of the science. And that is the recent Blade Runner movie. I don't know if you've seen it, but you probably don't need to know. Basically, the idea is you have replicants that are derived from human DNA and manufactured as adults. This is the character named Kay, who in this scene, a very artistic scene in the movie, I think, even though he's looking at this very anachronistic-looking DNA sequence machine, and this hologram of an AI wife substitute named Joy is reciting this very poetic line. Mere data makes a man, A and C and T and G, the alphabet of you and them, books made of sentences made of words, and all from 4 symbols, where I am only 2, 1 and 0. So my question is this: if mere data makes a man, and all you really need is sequence of DNA, why are we so far away from just going from the sequence of DNA to making an adult human? I would argue that we're much closer to the likes of a hologram such as Joy, who's probably just a few major technological steps away from our current Siri and Alexas. But despite all of the advancements in DNA sequencing technology, DNA editing, in vitro fertilization, and even the medical advancements that now can save premature infants, no one yet has figured out how to get around this step. And that is, as mammals, humans are manufactured inside the body of another of our species, and that is the pregnant woman. And what's becoming increasingly evident in today's medical era is that really the 4-letter code may only truly make sense when interpreted in the context of the maternal environment. And for all of your life stages, obviously gene-environment interactions are important throughout your whole life, but really it's increasingly seen as that first 9 months spent in utero probably has the biggest impact of the environment that will shape the rest of your life throughout all of your life stages. And particularly for gene-environment interactions, when again this set of instructions is being read, but in the context of what's inside the mother's body and in her environment, including diet, the nutrients in the diet, chemical pollutants, psychological stress, microbes such as viruses and bacteria, as well as the medications she's taking. So as an analogy, you can think about a contractor building a house from a set of blueprints, right? And there's probably many mistakes in the blueprints and inconsistencies, ambiguities. And if he had all the time and money in the world, he'd be able to come up with fixes and creative workarounds, right? But the realities of the outside stresses of the world means he's probably going to have to cut some corners on the final product, much as what's probably happening in the maternal body that's exposed to a lot of these stressors. And the builder obviously is going to prioritize the most important parts of that house, the weight-bearing beams, the roof, the floors, the windows, and maybe cut corners on the more luxury items in the house. Well, unfortunately, for the higher order parts of the human brain, including social behavior and language, evolutionarily, these are seen as luxury items. So now to bring it back to the state of the science, really all current evidence points to prenatal risk factors for autism spectrum disorders. And top of this list is what you just heard a nice talk from Dr. Geschwind, is that autism has a strong genetic basis. And of course, these genetic variants are present at the very earliest stages. And even for those new de novo mutations, they're there from the earliest stages. But also from other fields, including developmental psychiatry, developmental psychologists, if you go back to early home movies of children that are later diagnosed with autism, they're able to detect signs very early. The first birthday party is a classic one, and they can always tell the child that will go on to develop autism. And then in brain imaging field as well, it's getting increasingly good at detecting very early signs of brain differences that are there at the very earliest stages. I'm going to be telling you more in the next few slides about the medical and environmental factors that have been shown to increase risk for autism, and these are overwhelmingly seen at either the in utero or very early infancy stages. And just to head off the questions that may come about the controversy over vaccine in autism, I just want to say that really the preponderance of the evidence, many rigorous conducting scientific studies, including an epidemiological study of over 14 million children, has failed to find a significant association between MMR vaccine or thimerosal and autism risk. So now to bring us to the state of the science, I've got a couple slides to talk about the non-genetic autism risks, and again over here sort of summarizing in this sort of plus symbol as the strength of the evidence in the current literature. For medications, the big ones are thalidomide, which was used in the past to prevent premature delivery, and valproate, which is a mood stabilizer. These are sort of not much used anymore, and the current concern is the SSRIs, like Prozac. There's still sort of conflicting studies that it needs to be further studied. There doesn't seem to be much risk with the ovulation-inducing drugs used in fertility clinics, and there's really not any link now to ultrasound use. For infections, the big one is rubella infection. If a mother were to get a rubella infection during pregnancy, it increases the risk of autism 50-fold, which again is not so much a concern right now, but if you think about this generation of kids that didn't have their MMR vaccines as they grow up to be of reproductive age, it may start to be a concern for the grandkids of the parents who didn't immunize their children. For other immune challenges on the mother, having asthma during pregnancy or fever or inflammation are moderate risks, as well as influenza with a somewhat milder risk. Again, any sort of immune challenge could be involved. The metabolic conditions, these are ones that have increased in recent generations because of the obesity epidemic. The big one is having gestational diabetes really increases the risk for autism. Obesity itself is only a moderate risk, but this also predisposes to preeclampsia and hypertension during pregnancy, or also it's more the gestational weight gain. If a mother starts out as being overweight and obese and she gains more than the recommended amount, that will increase it more than obesity alone. Additional non-genetic factors, the big ones here are increased age of either the mother or the father are quite strong risk factors. On the chemical side, again, there's still more research being done on this. Probably the two most convincing so far for the largeness of the populations that have been studied are pesticides and air pollution that increase risk. And these are easier to do because you can look in larger populations from surrogate estimates based on geography, where the ones that are actually measured in women's bodies, flame retardant chemicals and phthalates have some emerging degree of risk. Some of these have a time-dependent, so season of conception or birth seems to play a role, as well as the inter-pregnancy interval. So this is the time between two different births from the same mother. And both of these are probably related to the nutrient storage in the mother. And that brings us to the protective factors shown here in the blue font. And the big one here is actually folic acid, which is the major component in prenatal vitamins. So if taking prenatal vitamins prior to conception, which is recommended to prevent neural tube defects, is also protective for autism. And then additional protective factors include iron in the mother's diet, as well as breastfeeding the infant. But it's important when we think about genes and environment and autism to not run the tendency of trying to put them in two separate boxes, because they really interact in what I like to call a two-way street interaction. And that is that genetic differences can influence environmental susceptibility. Environmental factors can also directly or indirectly damage the DNA. And then last but not least, environmental factors influence gene expression through long-lived epigenetic changes. So epigenetics literally means on top of the DNA sequence and refers to modifications on the DNA or the proteins that the DNA is wrapped around within the nucleus. And you can see that they're really well poised to be that interface between common genetic susceptibility and common environmental exposures that all may interact together in the disrupted neurodevelopment seen in autism. DNA methylation is what I like to call the first layer of the epigenetic code, and it literally just means these methyl groups, CH3 entities added on the mirror images of these CG sites, and these are throughout the entire human genome. DNA methylation can integrate genes, environment, as well as child outcome. And unlike gene expression, which is transient and in the moment, DNA methylation can reflect past exposures and potentially predict future outcomes, and is stable and measurable in human stored tissues. The unique challenge for epigenetics, which is not true for genetics, is that tissue matters, tissue type matters. And this is because if you think about all the cells in your body, they all, you use the same DNA set of instructions, but every cell is using those instructions differently. So the tissue type that you take that DNA sample from, it's going to matter when you do an epigenetic study. So obviously, for autism, the tissue we would like to sample is the brain, but you really can't biopsy the brain of a baby. So fortunately, there are two other tissues that are part of the normal birth process, and that is the placenta and the cord blood. Remarkably, these precious tissues for several past decades have been treated by the medical establishment as trash and thrown out with medical waste and thrown out with the trash. But I think it's being increasingly seen that placenta and cord blood are not only great accessible tissues that should be studied, but they're essentially time capsules of gene by environment interactions that could be really useful for understanding disorders such as autism. And so our team of collaborative researchers have decided to take on this challenge of seeing if perinatal tissues could be used to mine for epigenetic biomarkers that may predict autism from birth. And so the idea here is if you could come up with a panel of gene loci where percent methylation together could help predict if this child was at high risk for autism. There's some great behavioral interventions you'll probably hear about the next talk, that the earlier you start, the better. So again, identifying who would benefit from those behavioral therapies, as well as you heard in Dan Geschwind's talk, that knowing these genes and the gene pathways that are disrupted can help targeted therapies, as well as understanding the impact of environmental factors on specific gene loci that are relevant to autism could help in sort of policy decisions that could potentially prevent future autism cases. So I just want to briefly show you a couple glimpses of the basic science studies that back up our premise behind this approach. First, hopefully you can see here that this red line looks very different from the other tissue. So the red is placenta. This is percent methylation across a whole chromosome. And placenta is very unique from other tissues, including many of the brain tissues, in that it has these valleys of lower methylation that are called partially methylated domains. And when we looked across 8 different mammalian placentas, what we learned from that is what is really conserved, is that higher methylation over genes actually can predict gene expression in placenta. And this rule was actually more similar to oocytes and pre-implantation embryos and is not seen in the fetal tissues once they are implanted. So to apply this to human autism risk, I'm fortunate to be working with a great team of epidemiologists and clinical developmental psychologists at the UC Davis Mind Institute in this study called MARBLES, which stands for Markers of Early Risk in Babies Learning Early Signs. This study recruits mothers who already have one child with autism and are planning another pregnancy. And that, therefore, we get a recurrence rate of it's running about 22% of the samples stored from MARBLES will have end up being with a diagnosis of autism. So what's great is a prospective study is we can collect biological samples for the mother during pregnancy, the child before the final diagnosis, as well as the cord and placenta at birth. There's also questionnaires given to the mother about environmental exposures and food frequency, and then the clinical assessments are done actually throughout age 1, 2, and 3. We go with the final diagnosis at age 3. This study I call a lesson in patience because we're still probably this year going to have the number of samples to actually look for indicators of autism risk. But I just wanted to give you a glimpse of some of just our early results from the first about 40 samples that we collected. And that is, so this is our basic approach. We use a chemical called bisulfite that we can then use to mutate the DNA and detect where the methylation was, and this is followed by next generation sequencing. Here is basically every line is a different placenta, and hopefully you can see that everybody pretty much has the same overall landscape, but you can see those valleys, there's a bit of a difference. And when you turn the graph this way, you can see that there's more variation over those valleys as partially methylated domains. And again, this early study, we looked at the questionnaire data, which they had early on, and we saw an association between women who said they used pesticides on their lawn and garden and overall percent methylation over these partially methylated domains. So in the future, we're going to continue with this. We're getting better at getting this down to single gene loci and multiple more samples. We're doing a cross-site replication in collaboration with Johns Hopkins, who has a very similar experimental study design called EARLY. We're going to be doing both differential methylation with 2 different assays, combining this with genetic information, as well as measured exposures from the biological samples collected, and then the nutrients as well from these biological samples. So the idea is we'll be able to get a much better understanding of gene by environment interactions, as well as hopefully come up with a panel that could potentially be used for predicting autism risk from birth. So I'd like to sum up with this by having you imagine the future of autism prevention, diagnosis, and treatments. And again, my goal here is really not to blame mothers, but to value and help them. We certainly don't want to go back to the middle of the 20th century, where the refrigerator mother theory of autism was blaming mothers for being cold and distant. Clearly now, there's a clear, strong biological basis for autism. As you heard from the prior talk, there's great technological advancements in genomic sequencing that have brought us to hundreds of autism genes and hundreds of animal models that can really study mechanisms as well as those gene-environment interactions in a mechanistic way. So in the future, hopefully we could use this information to affect medical and societal policy changes that really value that period before and during pregnancy as the most important time for healthcare, to identify at-risk infants and intervene early and often, so that you can improve life functioning and life skills for children and adults with autism, and certainly work towards improving treatments based on the information found from these genetic and epigenetic studies. These are some of the, briefly want to show you the NIH Supported Research that's going to help us get to that imagined goal. So the epigenome roadmap has been underway. It's a great reference map of all the different epigenomes in the human body. Psych and Code is one that's specifically focused on the brain and neurologic disorders, including autism. Brain Initiative also will help greatly in the understanding of the brain. ECHO is a epidemiology-based study which is now underway to look at environmental influences on child health outcomes. This will be collecting cord and placenta samples from hospitals across the country. We still need those investigator-initiated research projects as well to go after specific hypotheses using all of these great resources. And with that, I'd really like to thank you for supporting basic science research. And I'd also like to thank my wonderful research team, our funders, which are all NIH public support, and my collaborators at UC Davis and collaborators at Johns Hopkins. Thank you.
Dr. Dawson, thank you very much.
Well, I also want to begin by thanking the AAAS and the Dana Foundation for this opportunity to give you a whirlwind overview of treatment of autism in 15 minutes. That's quite a tall task, actually, but I shall do my best. One of the reasons why it's so challenging is because there is so much heterogeneity in autism. So a person with autism could be an individual who has no language, who needs 24/7 care, who is struggling even to have a job, Or it could be the students at Duke that we provide support for. It's students on the spectrum attending Duke University who are struggling with some of the social impairments. But despite all of this heterogeneity, there are sort of two features that are common. One is a difficulty with social interaction, and that can be very fundamental. So a very impaired person may not even want to be interacting with another person at all, or have very primitive skills in interacting. A very capable person may still have profound difficulties in reading emotions and social cues. And of course, our world is so dependent on those skills that they also struggle. And then the second category is the presence of what we call repetitive behaviors or restricted range of behaviors. This could be preoccupations, ritualistic behaviors, favorite activities, and an overly focused interest in certain topics or objects. Now, I wanted to begin with the big picture, and I think this is particularly important when we think about it from the point of view of parents and what they are faced with as they think about putting together a treatment program. So right away, we often involve many different disciplines in the treatment of autism. So for example, autism is associated with a wide range of medical comorbidities. This could be sleep difficulties, seizures, GI disorders, food sensitivities, many other conditions that actually are in the psychiatric range. So as people grow older, they may have ADHD or anxiety, depression. So it's not just the autism-specific interventions, but these medical comorbidities that parents are often seeking for the individual with autism. And in fact, these medical comorbidities often have more impact on quality of life than the autism itself. And unfortunately, they're often missed because a child with autism, adult with autism, is not very good at describing their pain, and it can manifest in behavior. The physician may just interpret that as this is part of autism and really may miss these really important features. But we also involve the occupational therapist because there's difficulties in motor functioning and in the use of just objects and toys in everyday life. And then also a speech and language therapist is involved. So those therapies go throughout life. Early on, we also engage in early behavioral intervention, and I'm going to talk in more detail about that in a minute. And then on top of that, as we move from early intervention into school age and into adulthood, there's a range of other treatments that parents are seeking out for their children. Cognitive behavioral therapy is very helpful. For example, if you have a child with anxiety or depression or other problems with social skills that can be dealt with more of a kind of cognitive behavioral approach. Social skills training has been shown to be quite effective, and this can be done in a group setting or in the school context. Many children have academic tutoring because autism is often associated with a learning disability, and reading or arithmetic or writing can be particularly difficult. And then positive behavior support. So help with some of the behavioral challenges that may come with difficulties in, say, transitioning from one activity to another. So now keep in mind, and I'm still going to layer on a little bit more, is imagine now you're a parent who is actually interacting, right, with all of these different clinicians and taking your child to all of these different therapies. Many of whom may not even be speaking with each other, so it's really up to the parent then to try to put this together into a comprehensive program. So one of the movements in the field is to try to help parents really navigate this complex set of therapies that most people with autism need, and to try to come up with more of an integrated and collaborative approach across disciplines. So it's extremely helpful if the GI doc, for example, knows what the behavior therapist is doing, or if the psychiatrist is talking to the neurologist who may be assessing sleep difficulties. And then as we move into early adolescence and into early adulthood, it's very important that we focus on what we call transition planning, because what we've learned is that the period from moving from high school into adulthood is probably the most kind of vulnerable period, because you're moving from a point where there's a lot of systems in place, support, even if you think about just school, being able to take your child to school, to not having those supports. And interestingly, individuals who are more intellectually capable actually have more trouble during this period, although all, I think, individuals have trouble with that transition. And that's because there's very few support services for a person with autism that doesn't have an intellectual disability, and then they may have struggle as they move into college. So we know that it's really important to start transition planning and learning skills that will help the individual function in the real world, and that should start really around age 12 and 13. So for example, we found that having experience with a job while you're in high school, where you can learn things like, you know, how do you do an interview, and how do you navigate some of the social aspects of being in an employment setting? How do you take care of yourself in terms of hygiene? And so all of these are skills that take time to learn. And so the goal is to begin that during the adolescent and early adult period. And then throughout adulthood, again, all of these services are needed to be able to help the individual to function out in an employment setting, to develop friendships, and to also deal with some of the common psychiatric comorbidities that we see emerging in the adolescent and adult period, which can be, for example, anxiety and depression are very common. So suicide risk for individuals with autism is quite high, higher than many other disorders, as an individual is struggling and trying to navigate the social world as they move into adolescence and adulthood. So that's the big picture. Now, I want to talk a little bit about early intervention, and if you're familiar with the autism world, you may know that the most common form of early intervention is something called applied behavior analysis, or ABA, that's based on a technique that Lovaas developed that uses simple principles of learning, operant conditioning to help train and shape behaviors. And what we found is that this was largely successful in helping children learn skills. And this was quite a breakthrough, because we didn't think of autism as treatable before those early studies were done. But what we've learned that was kind of missing in some of those early forms of ABA is a focus on the social and affective relationship. Because what neuroscience and developmental neuroscience has told us is that the affective and emotional relationship is very key in drawing the child's attention to the social world. And without paying attention and having interest in the social world, learning does not occur. So because of that insight, many different interventions have been developed now that fall into a broad category that we call naturalistic developmental behavioral interventions. So it still uses the principles of ABA, but these are delivered now in a context that focuses on the social and emotional relationship between the child and the parent, or the child and the therapist. And we're finding that when we deliver the same kind of content in this sort of play-based and emotionally affective context, that learning is faster, and it's also retained for a longer period of time. A key aspect of this, too, is involvement in parents, because learning occurs throughout the day, right? Every moment of a child's life is a moment of learning and interacting with parents and other people around that child. So it's very important that we don't think of therapy as something where we take the child off and do the treatment, but rather teaching parents strategies that they can use throughout the day in every interaction that can promote social interaction, language, cognitive development. So there are many examples of this. They've been shown to be effective. One example developed by Sally Rogers and myself is something called the Early Start Denver model that uses this approach. And we conducted a study that was published in 2010 just to show the impact of providing these interventions very early in life. So the children began the intervention below age 2.5. And then we provided this intensive Early Start Denver model for 2 years. It was a randomized control trial, so children were either randomized to receive treatment as usual in the community or the Early Start Denver model. And what we were able to show was a very significant impact on IQ and language, adaptive behavior, social behavior. So the average IQ gain after receiving 2 years of intensive, and by that I mean about 15 hours a week provided by by therapists, excuse me, that the average IQ gain was 17 points. So that's over a standard deviation, has a very big impact. Now, we were also able to show at the end of the intervention that some of the differences that we in our lab had previously found in terms of patterns of brain activity that are associated with autism had now been normalized. So what we know in young children with autism is that they're not processing social information in the same way as a typical child. And you can see this as early as 3 to 6 months of age. So for example, early systems that underlie our ability to recognize faces and facial expressions, these emerge at about 3 to 6 months of age, and we can measure these using EEG and what we call event-related potential, where we just show a child a face or a facial expression and look to see, for example, whether they respond more to a face than an object or more to a mother's face than an unfamiliar face. And you can see differences in the way that the brain is responding by 6 months of age in a typical baby. So what we know in autism is that by 6 months of age, there's already differences in these brain systems that underlie very basic aspects of processing of social information, so the response to a face or a facial expression. So what we did is at the end of this 2-year intervention, we brought all of the kids back in, the kids who had received the interventions, the kids who had not, and then we also brought typical kids. And what you can see here is that the children who received the Early Start Denver model had patterns of brain activity that were virtually indistinguishable from the children with typical development. And what I mean by that is that now they were showing a brain response when they saw a face that was very similar to a typical child. So this was important because it showed that autism is involves a lot of neuroplasticity, and that perhaps what we see in autism by, say, 18 to 24 months of age is the downstream effect of a child not paying attention to the social world, and so they're not getting the typical kind of social input that you would have in normal development. And we know that this social input is very critical for early language and social development. So the idea is that if we can find autism early and provide that kind of stimulation, perhaps we can not only change outcomes in terms of behavior, but actually alter the course of brain development. Now, we followed children in this study for 2 years after the intervention, and we assessed what kinds of, when the parents just went out and sought any kind of treatment they wanted for their child, what kind of treatments did they receive, and how intensive were those treatments. And what we found was that the children who had received 2 years of intervention now were seeking out far fewer services in terms of occupational therapy, one-on-one therapy, speech therapy, and they were also much more likely to be in a regular classroom. And we did a cost-benefit analysis, and we showed that on average now we were saving about $19,000 a year after having provided early intervention. The estimate is that over a lifetime, that if you provide early intervention, that you'll save about $1.2 million for each individual in terms of not needing as many services. Now, we were able to show that the early intervention effects on IQ were maintained at age 6, and now we also saw a reduction in autism symptoms. So we were very pleased to see that the early intervention seemed to be sustained even after the intervention was stopped. Now, our goal at this point is not only to now get these interventions out to many more children, because there's still huge problems with access, not only in the United States, but worldwide. But our goal is to detect autism even earlier, because as Dr. Geschwind was pointing out, the changes in the brain are happening during the prenatal and early postnatal period. And by the time we're seeing symptoms, we're far down the developmental trajectory. And what we would like to be able to do is to detect infants right when those symptoms are beginning to emerge, or perhaps even before they are symptomatic, where we could provide some kind of stimulation and perhaps have a protective effect. So you can see, this is that home videotape that was referred to, and you can see the difference at 1 year of age at a first birthday party, and how a 1-year-old is responding to the cake versus the people around him that are singing happy birthday. So the typical baby is seeing all the faces and smiling, whereas the 1-year-old with autism is already focused on the plate and is not getting all of that social stimulation that we know is so important for brain development. So currently, this is a huge area of research, and I could spend at least an hour on any one of these topics, but people are trying to determine sort of biomarkers that could be seen very early in life that could tell us that an infant is at risk for autism. Some of these are behavioral biomarkers, so looking at how the baby's using their attention. Do they pay attention to faces? Others of these are electrophysiology, so we showed in our lab that by 6 months of age, that brain response to a face is already different. This is before the symptoms are even present. Neuroimaging has been incredibly promising. There's some beautiful work now following large cohorts of infants who are at risk because they have an older sibling with autism, and then neuroimaging the child prospectively. And what we're finding is that by 6 months of age, you can already see differences in the what's called the white matter fiber tracts. These are the tracts that connect different parts of the brain, as Dr. Geschwind was saying. And there's also some other early indicators that have to do with the extraaxial fluid in the brain that seem to be very predictive of developing later autism. And then, of course, the world of genomics and exome sequencing and proteomics, gene expression, all of these areas are very promising for early detection. So just to give you a flavor for what we might do in the future, what we did in this study is these were all infants at risk for autism because they had an older sibling. So we know that 1 in 5 children on a population basis, if they have an older sibling, will develop autism. So if we already have 1 child in the family with autism, the risk for the second child is about 20%. So by following these infants, we can prospectively see the early emergence of autism. So in this study, this was a large sample of high-risk infants because they were siblings. And then regardless of whether they had symptoms or not, at 6 to 9 months of age, they were randomized to either just be assessed as normally they would be, or they were provided stimulation. So the parents were taught and coached to provide social stimulation at home. And so this was a 9-week parent coaching treatment where they were taught how to really read subtle cues and provide extra stimulation that you typically wouldn't necessarily provide to your infant. What we found was that we could alter early patterns of attention, so now the babies were paying attention more to social information, and we also were able to show changes in that brain signal that we measured with electrophysiology, where now the baby's brain was responding more robustly to a social stimulus than a non-social stimulus. So this was a proof of concept study that we could perhaps identify infants very early, provide early stimulation, and again, change these patterns of attention and patterns of brain activity. Now I'm going to end my talk by, in the area that I'm mostly focusing on. I'm still very involved with early behavioral intervention, and our focus is on tailoring these more to different kinds of kids, but also on how you disseminate them. But we also know that despite receiving intensive intervention, a substantial percentage of people still have profound difficulties in social and language intervention. So about 30% of individuals with autism are unable to speak, for example. So we know that early behavioral interventions are not going to be the full solution. They will be all that is needed for many children, but many children are going to need much more. And so currently we're focusing on, could we develop drugs or medicines that could help to enhance neuroplasticity? And you know, right now there are no FDA approved drugs that address social communication or language impairments, which are the core symptoms of autism. But as Dr. Geschwind pointed out, because we are starting to better understand the genetics, we're now able to see that there are common pathways that these genes are affecting, and these are then pointing to drug targets that may enhance neuroplasticity. So a major challenge, however, is in conducting these clinical trials is trying to develop homogeneous subgroups for clinical trials. And the focus here is to develop biomarkers that could assess brain activity or patterns of attention with eye tracking that could then help to stratify individuals for clinical trials, or perhaps determine whether this drug is working very quickly by assessing brain changes or by assessing attention. This is a large study funded by the NIH that is validating these biomarkers based on either EEG and eye tracking. And then I want to end, this is my last slide, by saying that, so this is just an example from Duke University, because this is a very strong focus in my career now, is to collaborate and to provide the outcome measures for these clinical trials. But currently we're conducting 5 clinical trials that are testing different compounds, either molecular or cellular therapies. And some of these are focused on the reward systems, such as dopamine and oxytocin. Others are focused on the neuroinflammation, such as the mesenchymal stromal cells or the umbilical cord blood. Another one is a stimulant, again, focused on dopamine, the reward system. But that just gives you a sense that these trials are really not just an imagination, they're actually occurring, and we can discuss, if you want, some of the outcomes that we're finding. Thank you.
Thank you so much. Can you stay up here?
Sure.
And Dr. Geraldine, Dr. LaSalle, if you can take a seat. What we're going to have is questions, maybe answers. Here's the way we're going to work it. If you have a question, please come down the aisles to the microphones. I'll then be picking alternate sides. And I'm going to ask the panelists to pass around the microphone, this microphone, so I can use the ones up here. And I'll just start there. I think you were first, but.
I think I do have to take exception to a couple of things you say in your program. One is that the incidence rate of autism is about 1 in 110 kids. The CDC figure released just last spring was 1 in 50. And since boys are 4 times as likely to get it as girls, it's 1 in 31 for boys. Just think of that, folks. In every average 2 elementary school classes, you're going to have 1 autistic boy. This is a social catastrophe that we just don't talk about in the public forum. Secondly, you say...
excuse me, do you have a question?
Yeah.
Okay.
Secondly, you say that the idea that vaccines cause autism has been debunked. In the Republican presidential primary 2 years ago, the 2 doctors who were running both suggested that it would be a good idea to space out our vaccine schedule.
I know one of the, excuse me, I have to interrupt you. I know one of those doctors, Ben Persley, if he's the one you're referring to, Ben Carson. But,
Ed Rand Paul.
Great. But We're not here to debate some of that scientific evidence. I'm asking, do you have a question? Really?
This is a preface to a
Okay.
question. I think it's.
By the way, in fairness,
I think it's probably
I have
of interest to your audience.
I have to also state one thing I may not have stated. We have to end here at 7:10. There'll be a reception afterwards, and people can ask questions and make comments there. But could you get to the question? I'd appreciate
right.
it.
The same folks who've been telling us all along that vaccines do not cause autism didn't even know the glymphatic system existed till a few years ago. And the glymphatic system is intimately associated with the immune system, which is obviously related to vaccinations. Where's the question here?
Yeah, that's
How bad does it have to be before this is declared an epidemic?
Does any of you want to address that?
I think we can, I mean,
Could you pass the mic?
the problem is, and I think it's been mentioned, that the diagnosis for autism has changed, so it's really, you can't scientifically go back 20, 30 years and say that autism is, the epidemiologists would agree that it's not an answerable question.
Although when there have been 2 studies published with retrospective chart reviews, one in Scandinavia, one in England, where they blindedly applied the current diagnostic criteria, and they find the same rate as we do now. We agree. I think all of us up here, we've dedicated and are dedicating, hopefully in the future, the majority of our careers to trying to solve this issue. It's an enormous unmet need. And whether or not we call it an epidemic, whatever helps it move forward and focuses the research on areas that are most likely to provide improved diagnosis and treatment, that's what we're all about.
I mean, do you want me to?
No, you can.
Well, what I was going to say is that I understand why parents ask that question, right? So I think it's a perfectly reasonable question. And yes, the immune system is involved. Yes, it is associated in time. But when we have really, as scientists, tried to look at, with an open mind, right? And see, well, let's look and see if there's a connection. So far, the evidence just is not pointing that direction. So I'm an agnostic. I'm a scientist. I don't a priori make a decision about what is going to turn out. And I've looked at the data very objectively, and just so far we haven't been able to draw the conclusion. I think it's a very reasonable hypothesis, but so far no evidence has supported it.
Okay, one more follow-up.
The critical issue is that there is actually quite a bit of room for environmental effects. We know that there are environmental things that impact and increase the risk for autism, and Dr. LaSalle talked about potential gene-environment interactions, which we haven't identified yet, but which are likely to be occurring.
But I'll just comment. I'm a parent. Oh, sorry, I didn't know you.
Yeah, but we need to be, we need to ensure that we're actually studying the things that are likely to be those environmental things that we can remove. Nothing has been studied more than vaccines, and it just doesn't. It doesn't add up at this point. In multiple countries, multiple different methods. Anyway.
You hold on to that. I've got my own. I'll just comment as a parent myself. It's not just an epidemic, it's 100% disaster. So I'm with you that it's a big problem because it's really right on top of us. And with all the people I know, people like these here are working to try to find out the many different avenues that are responsible for autism because it's not one single thing. And with that, let me turn to you, sir.
I'm a high functioning on the spectrum, but out the closet. I'm just trying to find out, I want to confirm yay or nay, if you've taken imagery, computer, CT imagery of autistic brain versus neurotypical brain, do you notice any distinct differences, especially in more developed children or adults?
Yeah, I mean, the general answer is yes. Although again, we really need to conceive of autism as the autisms. And so there isn't a single universal finding, but there are a bunch of things that have been consistently found over and over again. Jerry, maybe you want to?
Yeah. So as I mentioned in my talk, I think one of the really interesting and important studies going on is a study following prospectively infants at risk and then using MRI to look at differences in the brain. And what we know is that what we call the white matter, so this is like the tracks, right, that connect, the cables that connect different parts of the brain. Particularly these long distance cables seem not to be developing normally. And if you think about what social interaction involves, so for example, right now interacting with you, I am using my vision to look at your face, I listen to your sounds, I'm interpreting your gestures. So we have to use many different parts of our brain in precise coordination in order to socially interact and use language. And so when those long distance cables between one part of the brain and the other are not developing normally, it disrupts these very complex functions of language and social interaction. So that seems to be what's the case. Now, kind of the good news, I would say, is that we're also finding that the connections within one region seem to be better developed. And that may be because there's strengths in things like memory or sometimes pattern recognition or music. And so we also know that autism comes often with great strength. 60%, there was a study just recently published, 60% of people with autism have some special strengths. And so it's, there are strengths and also these weaknesses, so to speak, that come from the lack of the long-range connections developing normally.
Can I make a corollary statement or question, which is, I don't know if it's always appreciated that when people talk about these studies of the neuroanatomy of autism, they're often typically talking about group studies. And the question is, can you identify an individual unequivocally? And
Yeah, so there is no MRI test, right, where you could say, yeah, I'm going to do an MRI and I will determine that this person has autism and this person doesn't. We're not at that stage.
I want to make that clear to everybody that we're not talking about individual diagnosis, the targeted yet. Sir.
Three of us spent the day today at another autism session at National Academy of Medicine, and the focus there was on non-human primate studies and genetic modifications. Not yet using CRISPR, but certainly looking forward to that, but otherwise using viral manipulation to turn off certain genes like CDH8 or something like that. We also learned that China, there was a representative from the Chinese Academy of Sciences, and he announced that they have recently initiated a major study closely working with Japan, but open to collaboration with anyone in the world, because they have the same problem, of course. As you said very clearly, this is a global situation. What I'm wondering, and my question, I do have a question, and that is, how can you as scientists, and more problematically, we as the public, keep up with all of the things that are in works to try to alleviate the terrible pains that people are suffering with autism? It just seems overwhelming.
I have a suggestion, actually. So I think this is open to the public. I think one of the best sources is a source called Spectrum News. Right? So the Simons Foundation has a subsidiary, so to speak, that is a news organization. It's called Spectrum News. So if you Googled Spectrum News Autism, it would come up, and they give a daily, very readable for the public overview of any of the new science that's happening. And I frankly use it to keep up, and then if I want to drill down into a paper, I do, because there's the links to papers from every one of those articles.
I'll comment also, though, as a parent too. It is overwhelming. So the slide that Dr. Dawson showed of all the different therapists and trying to coordinate that, I can only tell you my wife and to some extent me, we all went insane trying to run the child around and to appointments that to a normal child would not be as terrible as to an autistic child who at the mere thought of another DOCTOR, because we can't say the word out loud, would send him into spasms, jumping, and everything else. But I also have to caution everybody that we know that there's a gap between the science and the actual practice. And so you can read about these for interest, and Spectrum is one of the best sources for reading about it. New Scientist is another one I'll mention. I don't have an ownership interest in the magazine. I just subscribe. Has a number of interesting articles that tell you what's going on. But then actually translating the individual is, I think, a matter of the individual's concerns, their family environment, their capabilities, their resources. It's very different if you live out in the wilds of Montana versus if you're in Washington, D.C., for example, what services you can get. And it's also very different if your child is before the age of 21, and then after that, what we call the cliff of not having any services anymore. So you can keep up the reading, and I recommend it, but I think it has to be interpreted by clinicians and others as to what actually might make an effect for your particular individual. And sir.
Thank you. Everybody up there has mentioned how heterogeneous this autism is, and yet DSM-5 combined a number of individual syndromes into this one autism thing for some unknown reason. Is that really a step in the wrong direction? Shouldn't they be breaking it out again so you can actually look at the different syndromes as opposed to assuming everything's the same like cancer used to be?
Very good.
Well, being a neurologist, I'm not going to make any strong comments about DSM.
By the way, because people should realize DSM applies to psychologists and psychiatrists.
However, there is one of the advances, one could say, it may be 2 steps back, 2 steps forward, or whichever way you'd like to think about that, but one of the advances is that there is a kind of distinction where they talk about autism due to Fragile X, autism due to whatever the etiology. So there's a recognition there for the first time that autism has very, very heterogeneous etiologies, and that when one makes the clinical diagnosis that depends upon observation of behavior and cognition, you can also distinguish what the cause of that is. The hope is that that will eventually allow us to bring to bear more specific targeted therapies, whether they be behavior, combination of behavior and medications and other things. So without making any comments, so it does actually allow for that for the first time, specifying the etiologies, which I think is helpful with regard to the question you're asking.
Jerry?
So I actually think it does make sense to have this umbrella term, at least in this point in science. I think it may differ, in the future, which is the spectrum, right? That is the common feature is this difficulty in social communication, because it really we know quite a bit about the neurocircuitry. None of our lectures were on that. That's an area I've studied that underlie the ability to socially interact with another person. We know a lot about the brain regions that are involved in that. So you can have those brain regions involved in a person who, also has intellectual disability and a language impairment, or one of these specific genetic syndromes, or in an individual who's highly capable, maybe a professor at a university, but still those same, I think, neural circuits that allow us to interpret facial expressions, make eye contact, are involved. So there is some commonality. But as Dan was saying, when you make a DSM-5 diagnosis, you're also supposed to have all of these specifiers. Does the child also have intellectual disability? Do they have seizures? Do they have GI problems? Do they have early or late onset? Do they have a genetic etiology? So that's where the individual child is captured. and then you're put into this very broad category.
Thank you. We have time for at least one more question, hopefully one more. Ma'am?
What, if any, interventions or recommendations would you make for an adult with autism over 21 who's missed out on transition planning and so now has problems with staying in college, dropping out of school, and that kind of thing?
So one of the most important things is to work on employment. So there has been a number of recent studies that have shown that having some kind of job, and it can be part-time, and it can be, a very simple job, is key to, first of all, having something that is purposeful, something where you have a sense of accomplishment. You also have an opportunity, for learning a lot of skills, because being in a job setting requires social skills and whatever, job you're doing. And it always involves social interaction. So, and it is the most strongly correlated factor with mental health, is having a job. So I always think focusing on that is really key, and vocational rehabilitation services are really important in the as a service that is provided by the government that can help and should be able, if you apply for it, to provide a job coach and to try to look for services in the employment area. But there are many other things, and it depends on the individual and what the particular challenges are for that individual, which can differ. But you may have a comment.
Well, I'll have you can hold into the mic. I have my own. Okay. I'll also make a comment. It depends on where the individual lives. So, for example, in Maryland, there's the Developmental Disability Administration, and there's caseworkers that can be assigned potentially to people. And so they can help navigate some of this, and they also can help break down what I'll have to say are some barriers, potentially. So a lot depends on just where the individual is, and there are things to be explored that way. So in addition to the services a parent or concerned individual can do, there are professionals who might get involved even after age 21. And then Dan has.
Well, I just want to echo the job. I mean, my area is not treatment, obviously, but one of the things we recognized, even at a large state school like UCLA, we realized that we had hundreds of autistic students, most of them undeclared, but about 100 or so declared. And so we've started to develop evidence-based programs. First, we have to collect the evidence, but it's focused heavily on internships and jobs and these transitions. So I just want to echo that this issue of getting jobs and having employment, even if one isn't necessarily living independently, is really critical.
Thank you. And with that, thank you. Thank all the speakers.